Well, welcome to this talk. Now, I've just got off a video call with Dr. David Speicher, the Canadian DNA scientist and virologist, and I'm going to play the whole talk shortly, but I'm just going to give you a summary of it now for people that have got limited time. First of all, this paper is peer-reviewed, and already people are trying to get it retracted, redacted, taken down, whatever the word is. Yeah, retracted. Trying to say it's a non-valid scientific paper, but it is. It's a good scientific paper. So some people seem to be trying to get it retracted for reasons that aren't entirely clear. Dr. Speicher says there's teams analysing every sort of jot and tittle in the paper to try and find some justification to get it taken down, rather than debating the actual content of what it's saying. So that was disappointing before we even start looking at the science. So the vaccine vials are from pharmacists, including the XBB 1.5. So these are vaccine vials here that is collected, and they're analysing the amount of DNA contamination in these vials. And if we just look at that, maybe look at that top graph there in some more detail, we see that these are all the different vials. They've all got DNA contamination. Now, this was the old level of contamination that was allowed. This is the new level of contamination that's allowed. But of course, this is a log scale, so that's 10, 100, 1,000, 10,000. So basically now 1,000 times more DNA contamination is allowed than used to be allowed. Maybe more on that in a minute. So the DNA contamination in the Pfizer vials was 36 to 153 times that which was allowed by the regulations. That's not 153%, that's 153 times, 153 fold for the Pfizer. 36 to 153. With the Moderna, it was 112 to 627 fold. So huge amounts of DNA contamination compared to that which is allowed. And it looks like the Australian and Irish vials had even more DNA contamination. And the fact that the amount of DNA contamination varies in various places around the world means that the manufacturing process is clearly inconsistent. All Pfizer vials had SV40 DNA sequence, 500 billion, that's right, 500 billion copies of SV40 sequence per vial. Now, the thing about this SV40 sequence is, this SV40 sequence of DNA comes from something called the simian virus. But this SV40 sequence codes for a protein, and that protein inhibits the activity of P53, which is a human tumour suppressor gene. So if you're making a protein that's inhibiting the activity of the tumour suppressor gene, that tumour suppressor gene is no longer going to inhibit the tumours, therefore the tumours will be freer to grow. So that's a particular concern, and 500 billion copies of that SV40 promoter sequence per vial. Quite appalling levels of contamination, I think you would agree. The sequences that are there, he considers that the fact that there's so many sequences there make genomic integration more likely. So he said it's a bit like a shotgun or a shrapnel effect. The fact that there's so many DNA contaminations, it means that integration into the host genome, that's the human genome where it will cause a mutation, become more likely. Many DNA fragments, shotgun shrapnel effect. Small chunks can be spontaneously integrated into human DNA. And any that is integrated into human DNA will change human DNA. Therefore, that is a mutation, and mutations can cause cancer. The data we have so far is from VAERS, but David is concerned about more cancer cases, particularly going on for years, decades, and he's also concerned about harm to the human germline, that this could affect sperm cells, for example. We're not going to really know that for another generation, but there could be a generational effect. Now, normally I would think that's a bit scaremongering, but this is coming from a DNA scientist, so we have to take that warning seriously. Now, this allowance for the amount of DNA contamination, it was increased from this relatively low amount I think that was in 1996 that was changed by the World Health Authorization. It was increased to this much higher amount. So you can see by the old criteria, all the vials are exceeding it. And quite a few are even by the newer, more lenient criteria. But the problem here is that these guidelines were designed for naked DNA in vaccines. So if you just inject, so if you've got your vaccine antigen and you've got some naked DNA, that's just DNA fragments just floating around in solution. That's naked DNA. Then that's going to go into the blood. It's going to be got rid of by the body's natural defense systems, and very little of it will transfect, that is, go into cells, because the DNA is essentially water soluble. It's not going to go through the fatty cell membranes. So these allowances that we see here, these allowances were for naked DNA, which is remarkably inefficiently transfected across cell membranes into our human cells. But of course now it's in lipid nanoparticles, and the lipid nanoparticles carry the DNA contamination directly into our cells. So the fact that this DNA is now in lipid nanoparticles means that, I don't know, it's maybe 1,000 times easier, maybe 10,000 times easier for the DNA to get into our cells. Therefore, the limit should have gone down by 1,000 or 10,000 times rather than up by 1,000 times. That we should have a much more stringent requirement for lack of DNA contamination than we have, because it's transfected so readily. So the fact that this is above regulatory amounts is only a very small part of the story, because those regulatory amounts are designed for naked DNA. Now it's in lipid nanoparticles, and a tiny amount of DNA is going to transfect, get into our cells so much more readily than it did when the DNA was just naked in previous generations of vaccines. This is a real effect that simply hasn't been taken into account. So David's pretty convinced that there's transfection into human cells. Now, spike protein, of course, has been found in people 700 days post vaccine integration. So David thinks that because the spike protein is there 700 days after vaccination, that's because information to make the spike protein has been integrated into the human genome. So he said that's likely evidence of integration into the human genome already. Where it is it in the heart? Is it in the lymph nodes? We don't know. But it looks like our own cells are producing it somehow. Now, it's also possible that the pseudo-uridine, which is put into the RNA vaccines, he said it makes the RNA rigid and plastic-like. It could be that that's hanging around for a long period of time. So he can't say for sure whether the spike protein that's there 700 days post-vaccine is due to genomic integration. He thinks it probably is, but it could also be due to the pseudo-uridine. Either way, of course, it's bad. But this could be evidence for integration into human genomes already. And there's also the frame shifting as a result of the pseudo-uridine, which is bad. Now, David got his results on DNA contamination through two completely separate methods. I'm not going to go into the details. I don't pretend to understand them, but he does. Two completely separate methods, and they both show these high levels of DNA contamination. Due to now the amount varies because the manufacturing processes have varied, as we've said. And he said that pharma does measure the mRNA in their completed products. It does measure the DNA in their completed products, but they use different methods to do that, which just seems utterly ridiculous. Utterly ridiculous. His work has been sworn in by Senator Ron Johnson as evidence for the US congressional record. So even if, I don't know, say vested interest does succeed in getting David Speicher's paper retracted, whatever the word is, taken down, it's still been submitted to the US Congress as evidence. David, like me, calls for a moratorium on all these mRNA vaccines. He calls for a halt. So many people vaccine injured already. Let's not have any more. And any future mRNA vaccines, we need a lot more data before we are convinced that they are safe and effective, because the current ones aren't. He also wants access to full data so that people can have freedom of choice. Now, I did ask David, I said, look, if you were given some tissues from a human cancer, could you look into that cancer and tell me if there was any spike protein there? And he said, yes, he could. In other words, you can tell the difference between spike protein DNA and human DNA. And also he pointed out that the plasmids that they use as the DNA template for this are fully synthetic. The genome of that fully synthetic plasmid is actually in the public domain. Pfizer put it into the public domain. And David, if you gave him some tissue samples of human cancers, you could tell that that little bit sequence of the genome there was a human DNA. That little bit there was from the fully synthetic plasmid DNA contamination. Then the next bit was from human DNA. So if he was given the cancer samples, you could actually tell if DNA contamination from the fully synthetic plasmid or DNA contamination from the spike protein genetic information has been included into human cancers. That could be done. In our view, David's view and my view, it should be done. As far as I know at the moment, it's not being done. David is also working on the possibility of transfection into sperm cells. So that was a quick review of the interview. For those of you that are interested, it'll take a bit of time to listen, but I'm going to give you the full interview now. But that was the essence of what was said. So thanks to Dr. Speicher for that, and keep up the good work, my friend. So welcome to David Speicher from Canada. David, thank you so much for coming back.
John, it's always nice to be here.
And David is well known to people who watch this channel regularly. He's a DNA biologist and virologist and lab scientist of international renown, I think it has to be said. Now, we're here to look at your new paper, David, Quantification of Residual Plasmid DNA and SV40 promoter-enhancer sequence of Pfizer, BioNTech and Moderna modified RNA COVID-19 vaccines from Ontario, Canada, which is a bit of a scientific mouthful.
It sure is, I know.
Just before we go on to this, basically this paper is about the DNA contamination in the Pfizer and Moderna vaccines. Now, it's now published in peer review, but I believe some people are already trying to get this retracted because they don't like proper science, one must assume.
Exactly. There's a whole team out there of pharma paid folk to see things like this and basically examine every aspect they possibly can, every type of reference, everything. And so far, one of the biggest hang-ups is that in the abstract, I put World Health Authorization and not World Health Organization. And we all know they're self-appointed authority, which should be, yeah.
Very frustrating. So it looks like there's actually teams of people that are trying to undermine the scientific the process that you have gone through so meticulously.
Yep, it's all linked on the pub peer website known as PubSmear, but they say as soon as you are over the target, making a change and making a difference is when you will receive the most push back. So
Yeah, absolutely.
The facts are the facts. I'm a scientist. It's about the facts. We take vaccine vials, test it, and this is the outcome.
Where did these vaccine vials come from, David?
So a colleague of mine is a pharmacist and a very tired one, and she had pharmacists in her area donate them to research. And so most of these were basically old expired ones, except the XBB 1.5s, which because this work started in 2001 T3 to confirm the work of Kevin and so at that point, some of the vaccines were 2 years old, were in storage, that they're expired. So we took them to be looked at.
Do you believe these to be a representative sample of global supplies of Moderna and Pfizer vaccines?
Well, it's very hard to tell in full, because first of all, most of all of these are of the North American ones. I've also looked at vials from Australia and Ireland, and we're seeing more DNA in the Irish and the Australian vials. Except we do know they use the same manufacturing method globally, and therefore we know worldwide all of the COVID mRNA vaccines have high amounts of plasmid DNA, and all of the Pfizer's have a SV40 promoter-enhancer sequence.
So it seems reasonable to believe that there's variation in the manufacturing quality of the end product in various parts of the world.
There sure is. If you move to the next figure 2, as we say, the process is the product. And so all of this is for fragmented residual plasmid DNA. All of the first four Pfizer vials are the purple top vials, which are a PBS buffer. All of the other ones of Pfizer and the Moderna have a tris buffer. And so in that photo, all of the purple top ones, have a PBS buffer.
But all of them contain, I mean, the Pfizer ones contain between 36 times and 153 times more DNA than the levels, the maximum levels set by the USA Food and Drug Administration and the World Health Authorization.
Exactly.
And the Moderna vials had 112 to 627 times more.
Yes.
This is fold. This is not percentage. So some had 627 times more DNA than was actually allowed.
Our largest Moderna vial had 6,700 nanograms per dose of total DNA.
I mean, that means you're well into the microgram range.
Yes. And there's no SV40 full virus, as people have said. Oh, there's no actual virus in this, or the whole thing, except there is around 500 billion copies of the SV40 enhancer and promoter sequence, and we've shared this in the past.
500 billion.
500 billion copies of SV40 sequence in your files.
Wow, per vial.
Yes.
Right.
Yeah. Well, it's 500 billion copies per dose.
And the significance of this is that the SV40 DNA will actually produce a protein that inhibits the body's own tumour suppressor genes. Is that right? The P53 tumour suppressor genes.
Yeah.
Right. So in other words, we're giving the genetic instructions to make a protein that will inhibit the body's own cancer prevention strategies.
Yep.
Therefore making cancers more likely, because we're inhibiting the body's ability to defend itself against malignant cells.
As well, the SV40 enhancer, which is routinely used in a lab to integrate DNA sequences into a cell's own genome.
Right. So there's genetic instructions in the DNA contamination that will make integration into the host DNA more likely.
Yes.
Wow. And of course, that changes the DNA, and that makes mutation, and mutation predisposes to cancer.
Now on the Moderna end of things, there's no SVV40, except there's way more total DNA. Most of those are small fragments, and therefore, if you bombard a cell with small DNA fragments, it's like shotgun shrapnel into that cell, and those small chunks can spontaneously integrate.
Yep. Can they integrate without reverse transcriptase?
I don't know. I need to look. I think so.
Yeah, that's my impression. That's my impression. Now the orange graph here below shows the total adverse events from the VAERS.
Yes.
Now, okay, there is some correlation there with the vaccine doses, but those adverse events, of course, are going to be reported in a very short time frame.
Yeah.
Whereas my main concern with the DNA contamination is that over months and years and even decades, we're going to get more cancers. Is that a justified fear that I have, David?
Yep. It's all short term reverse events shown on VAERS, and I don't think we will know the full extent of harm caused by these genetic vaccines in our lifetime or our kids' lifetime, John.
Wow. So in other words, there could be effects here on the next generation.
Yes.
I'm disappointed to hear you say that, David, but if you're saying it, then we have to listen.
Well,
because you're the DNA scientist.
I kind of hope that I don't. I hope that I'm wrong.
Of course.
But there is work being performed on sperm cells to see if this is in a sperm cell and being passed on.
Yeah. In other words, germline mutations.
Exactly. Is it in the germ lines and being passed on? And if it's found there, then we've got a serious thing.
Well, it's bad enough with cancers in the current generation, but the idea of affecting future generations is pretty terrifying. This graph here, David, that we're looking at. So we've got the levels now.
Okay.
I think we need to make viewers aware that the first thing you do with a graph, as all our viewers know, of course, is look at the y-axis, and we see this is logarithmic. Mm-hmm. So it's going up 10, 100, 1,000, 10,000. So a little bit over the lines is actually an awful lot over the lines. But we see a dotted line at the bottom here, historic FDA 10 picograms of DNA dose guidelines. So in the past, the FDA only allowed 10 picogram range. Now the FDA allowance is 10 nanogram range, which we see above. In other words, they've reduced the level, they've increased the level of DNA contamination allowed by 1,000-fold.
Yep.
When did that change
Now,
take place?
It's very important and to keep in mind these levels are set for DNA vaccines and not mRNA and DNA in a lipid nanoparticle transfecting cells.
Yeah.
And it's for naked DNA.
So I think if we just explain that, I think make sure I've got this.
Sure.
So if you're injecting naked DNA, a lot of that's not going to get as far as your cell membranes because it's water soluble.
Yes.
Whereas if it's in a lipophilic lipid nanoparticle, that lipophilic surrounding is going to transfect it directly into cells.
Yes.
So these guidelines actually for naked DNA, which is water soluble and unlikely to get into cells, don't seem unreasonable. But I would have thought if you're putting the DNA in a lipid nanoparticle, these guidelines should be 1,000 times less, not 1,000 times more, at least.
Yes. Before 1986, the WHO set the lower level at 10 picograms per dose. In 1996, the WHO increased it to 10 nanograms per dose.
That's 1,000 times more allowance.
1,000 times more, based on speculative calculations. And then in January of 2003, the WHO granted plasmid DNA-based vaccines the privileged status of ordinary vaccines, and therefore we have a level set at 10 nanograms per dose.
Yeah. I mean, with the lipid nanoparticles and these levels, we've got guaranteed transfection, haven't we?
Exactly. It's just if you're making a king spike rotating 700 days post-vax, you've got integration occurring somewhere in your body. It's just it's very difficult to find and very difficult to show absolute certainty of what's going on. Is it in the heart cells or a lymph node or where, right? So
is it possible that the addition of the pseudouridine could increase the time the RNA can hang around in the cytoplasm of cells up to 700 days, or in your view, is that impossible?
Yes, absolutely. It makes the RNA very rigid, plastic like also makes ribosomal frame shifting, and therefore you're not just making a one-off targeted response to full-length spike.
It's the question I'm trying to get to here, David, sorry, is that if the spike protein 700 days post-vaccine, does that necessarily mean integration? Or could the presence of that spike protein 700 days post-vaccine be potentially attributable to the presence of the pseudouridine? I mean,
it's probably a mix of both.
Okay, so basically, you are strongly suspecting some level of integration.
Some.
Got it. Right. So specifics of the DNA multiple. So we've got basically you use two methods here. You've got specific DNA of multiple plasmid targets.
Yep.
And you've got total DNA. So the total DNA, we've got something like for the Pfizer, 371 to 1,548 nanograms a dose. With the Moderna, it's higher. So that's like different types of DNA, isn't it?
Well, it's total plasmid DNA.
Right.
Okay, and so after it's all grown up in a E. coli cell, it's all harvested out of those cells.
Out of an E. coli.
Yes.
Yeah.
Purified and then used to make mRNA.
Yeah.
The thing is, the way pharma digested that backbone, sometimes it was good and sometimes it
right
isn't, and so it chopped up things in various sizes, and our largest size found is around 3,500 base pairs or a full spike gene, and our average size is around 215. And so in Moderna, it's clear that Spike did not digest as well as the other parts of that plasma did.
Yeah. Now to work out the total amount of DNA in the vaccine doses, you use two different methods.
Yes.
So does that make you more certain that your results are correct? And were these methods completely independent of each other?
They were completely independent of each other. And what's quite ironic is that pharma will measure total mRNA based on free.
That's just a way of measuring RNA.
Yeah, and DNA based on PCR, and therefore you've got a mixed method thing, and PCR levels are much lower because you've got it's not all digested in the same spot, and you've got fragments of various spots and sizes and things and things, and so the image reads higher, and the EMA guidelines show that the DNA maximum is supposed to be no more than 1/3 of the level of mRNA. So,
and it's higher than that. It's nearer a half or 2/3, is it?
I bet it's about a third to a half, except if you take multiple methods to test things. It's a very good way to change or fudge your results, right?
So it's almost as if people could change their methodology to get the results that they wanted. Not that we're saying they do that, of course, but
exactly, yes.
But that could be done. And you also test for the specific sequences that we talked about. Yeah. So are you more the specific sequences that will code for the SV40? Are you more concerned about the total volume of DNA? Let me ask that again. Is the total volume of DNA a concern as well as the specific SV40 DNA that could inhibit the p53 suppressor gene?
I am worried about both.
Right. Right, got it. Now, that's excellent. I think that's the main points of this study. It's incontrovertible that. Go ahead, David. Sorry.
It's very big. It's there. The facts speak for themselves, and it was a great honor last week to have this work sworn in by Senator Ron Johnson as evidence on the U.S. congressional record.
Yep.
So it's there on record, which makes this a very important piece of science.
What should governments around the world do about the mRNA vaccines that are still being used?
Well, I would say call a complete moratorium on them, a call to halt, and we've seen so many vaccine injured that it's time to end this before more lives are harmed, John.
Yeah. Yep. Pretty short debate, I think, that one, David. But it's not being done everywhere.
Well, I think as well. There's so many out there which believe that it's safe and effective, that they need it, and I think as well that we need access to info, follow the science, and give people freedom of choice. If you don't want it, fine, cool. If you do, Alberta here in Canada, the vaccines are available. Except if you want them, you pay for it.
Okay.
And
so less people. The good thing about that is less people will be taking it up.
Yeah, and it's not taxpayer funding going to waste.
Good. So there's been some progress. What are you working on now? What's your next project, David?
Well, our next work is to examine a large database of vaccine injured people to work out how things have changed with CD4 counts, CD8 counts, all kinds of immunological markers, as well as the whole thing on genomic integration into sperm cells.
Yep. If you were given a tissue sample of a cancer,
Yep.
could you look into the DNA of that cancer and see if there was any evidence of spike protein integration or DNA contamination integration?
Yes, I could, John.
Because the DNA integration, you would be seeing E. coli DNA, not human DNA.
No, there's very little, if no E. coli DNA in these files, John.
Oh, right. But you could recognize the plasmid DNA to be more correct then. Yes.
Yes, it's a fully synthetic plasmid.
Oh, right. But you know the genome of that fully synthetic plasmid.
Yes, we do.
So right. Oh, so the E. coli are just the vector, just the piggyback. Yeah, the host.
Yes.
Yeah. All right. So fully synthetic.
It's a fully synthetic.
Right.
And two, it's out of Pfizer's Department of Gene Therapy.
Right.
And that is a Pfizer published paper.
Yeah, public domain.
Yeah, it's there. It's open and it says that this here is out of our Department of Gene Therapy.
Yeah. So basically, if you were given some human tissue, you could say, you know what, in that cancer there's some mRNA spike protein sequence.
Yep.
That will correlate with the development of a cancer. Or you could say, well, in this cancer here, we see parts of this synthetic plasmid DNA that we recognize the genetic sequence of. Therefore, that has been integrated into the DNA of this cancer cell.
Well, it's possible lab-wise, and to show that you must sequence that cells chromosome and prove human plasmid human.
Yep. Got it. Yep. So there'd be a human bit, then a plasmid bit, then a human bit. Yep. So really, you should be funded to be analysing cancer tissue samples from around the world, particularly, for example, bladder cancer that we believe is more highly associated with the mRNA vaccines.
That would be a very good thing if our world's governments would fund that research.
Well, I think it would be a good idea. I think it's a good thing for science. It would be a good thing for medicine, but I'm not holding my breath on that one, David. But
nope.
Yeah, that is excellent. We'll put all the links, of course, to David's materials. And we've also played a few clips of we've gone along of you working in the lab, David, which is also quite nice to see.
Cool.
Always good to see a man actually on the coalface, laying the bricks, testing the DNA samples.
Exactly.
Right, for now, David, do come on next time you have a publication. Of course, you'll be welcome with open arms.
Awesome, John.
We just need more proper, good, objective, non-partisan science. Science is the objective study of the nature of reality.
As I say, follow the science and the silenced.
I'm going to write that down. The science and the silence, yep.
The science and the silenced. If they don't want it out there, they'll
shut you up.
And shut you up.
They're trying to stick socks in your mouth as we speak, David.
Yep. Except I can't be bought, John.
Amen to that, David. Thank you. We on behalf of, well, as far as I'm able to speak for the ordinary people in the world, thank you for what you've done. Great stuff.
Thank you.
As always, David, God bless you, and thank you for coming on.
Thank you, John.